Right, everyone, thank you very much for choosing this talk. I'm excited to be here. So, today I want to tell you guys about our work doing genomic predictions at MyHeritage. And I have two main goals for this talk. The first one is to get you guys excited about doing machine learning genetics. And there's several reasons to be excited about that. One is the really unique data scale, which provides many challenges. Another one is the opportunity to really develop novel statistics, machine learning, or deep learning techniques which are tailored for genomics. And the third reason is really the potential to improve people's lives with good predictions. And the secondary reason for this talk is to give you guys a taste of our approach and just describe some of our methods at a high level. All right, so the talk outline, we have three parts. In the first one, we'll be talking about heritability and genomic predictions. Then we'll be talking about DNA and the data format, and then about methods and results. So before we start, let me tell you guys about MyHeritage just briefly. So MyHeritage is at its core a genealogy company. So it started off providing family tree services, as well as tools for people to research their family history and to find documents. And there's a lot of documents and a lot of tools for that. And since your heritage has a lot to do with your DNA, about 1.5 years ago, the company started doing DNA tests. And that's where the science team was formed. And the science team is doing several things right now. One is ethnicity estimates. So out of the supported ethnicities that you have, you might get an estimate like that with what percentage of which ethnicity you're from. Another thing we're doing is relative matching. So given your DNA, can we find people in our database which are share genetic segments with you? So it might be a long lost brother or a fourth cousin. And third purpose of the team is to do longer term research about genetics mostly. So cool. So let's get down to business. So we talk about heritability and genomic predictions, and let's start with what heritability is, and we'll talk through it by an example. Okay. So let's consider here Frank and Molly. Okay. So Frank and Molly have 2 kids, Ben and Lisa. And we know the following about Frank and Molly. So Frank is 185 and Molly is 175 meters tall. And Ben has high LDL cholesterol, okay? So that's the bad cholesterol. And Molly has a regular level of LDL cholesterol. So we might ask ourselves a few questions. So one is who here, for example, thinks that their son Ben has over 50% chance of being above 175 meters tall? All right, so most people, right? And me too. And who here thinks that their daughter Lisa has over 50% chance of having high LDL cholesterol? So not many people. I'm not sure. I think it's just because it's a harder question. And the reason why it's hard is because whereas height and cholesterol levels have a genetic component, they also have a very strong environmental component. So it's not just a function of your DNA. And this is perfectly illustrated by this cartoonstock.com publication, in which the lady's weight problem is partly explained by genetics and partly by Boston cream pie, right? And that's the core of the thing. So how heritable are these traits? And how could we measure exactly the percent of the genetic component of these traits? That's what we mean by heritability. So how can we measure heritability? There's many reasons why it's hard to measure heritability, and when I started learning about these things, I was quite puzzled. So there's many things that are hard to control for. So one is environment and nutrition, for example, right? If you're talking about cholesterol levels, there's all kinds of things that matter, like what you eat, right? That matters a lot. It's also hard to control for income and healthcare, for example. So someone that has more access to good healthcare will likely have different outcomes. And also prenatal effects. So that's what I mean by that is what happens during pregnancy. That also affects you a lot. So how could one possibly control for all these things? And the answer comes, and it's very widely done in genetics, and it's been done for decades already, and it's twin studies. So in twin studies, so let's talk about height, for example, here, okay? What twin studies can do is to compare perfect twins, which are twins that share 100% of their genes, against regular twins, which are twins that share 50% of their genes, okay? And and sort of like this like comparison controls for like all the things we were talking about before, like environment and nutrition. Like they likely ate the same things. They had access to the same like income and healthcare. They also like went through the same pregnancy, right? So like controls like even for that. And I'm not like going through the details here, but you can, for example, like compare like the correlation in height between like perfect twins against the correlation in height between regular twins. And depending on like how much higher it is, you can infer the heritability. So like I'm skipping the details for that, but hopefully it's clear that something like that can be done. And so these are some of the results for like how heritable these traits are. And these come, I think, all from twin studies as well. So I know there's like a bunch of things that really surprised me here. So like one is like how highly heritable like some psychological disorders are. So like I like used to think like out of like ignorance, I think, like many things I think, that these diseases were like almost like entirely environmental, but they actually have like a very strong genetic component. So for example, schizophrenia, which I think in the general population less than like 0.5% of the population will develop that. But if you have a perfect twin that has it, now you have over 30% chance of having it. So like it's a really strong genetic component. And we'll be talking here a lot about height, okay? And the reason we're talking about height is because it has a very strong genetic component. There's a lot of data for it, and it's also interesting because it's highly polygenic, meaning that sort of like all kinds of areas in your genome have an impact on height. So it's a really interesting example to study from a machine learning perspective because it's really hard. And another thing to note here is something called like the missing heritability. All right, so this is like a term like widely used in genetics, and what it means is that like most algorithms that try to predict these traits, they have not achieved sort of like close to that potential which is indicated by twin studies. So you know, like whereas like you can think of sort of like that in the right column as sort of like the best possible, you know, like R squared if you want to predict height. And so for height, for example, like as far as I'm aware, like the best algorithms have achieved about like 40%. So there is like really like ways to go when it comes to that. And yeah, I wanted like just to highlight this one more time. This is like really cool that these sort of like upper bounds exist, right? This is something which is not very common in like in machine learning at all. Like you can always, you know, shoot for, you know, like 100% accuracy, right? And good luck. But when you don't, when you know that this is not possible, you don't really know like how well you can possibly do. And these upper bounds, these like heritability estimates, they tell you like approximately sort of like how well you can possibly do with your algorithm, which is quite nice. Cool. Any questions here about this setup?
We'll take questions at the end.
Okay, sure. So, and so before we go into talking about the data and the methods, let's talk about just quickly like why we should care to predict these traits. Right, so like one is that it allows people to take proactive action. So the classic example here, this is like an op-ed in the New York Times called My Medical Choice. And it was written by Angelina Jolie, and she's explaining like why she decided to remove her breasts after she was diagnosed with an 87% chance of developing breast cancer. Right, so like this is not just from like, you know, the 87%. She consulted with like many doctors and did many other exams, but this kind of estimate like allows people to take proactive action. And this is like a really like extreme and unfortunate example, but there's like many other ones that, for example, like if you know you have like a high chance of developing skin cancer, you can like, you know, conduct yourself in a different way during life. Another reason is personalized medicine. So like, for example, pharmaceutical companies can understand that certain type of person will be able to, you know, react like extra positively like to a certain kind of medicine, or react negatively, like depending on their genetics, and these drugs can be prescribed more accurately. And the third example, which is a bit more sort of like futuristic, is CRISPR. So CRISPR is a technique for genome editing, and it was developed like only like in the last few years, and it pretty much it sounds it's like as impressive as it sounds. It seems to allow for like people to edit specific parts of your DNA, and this like has not been done to a meaningful extent in humans, but you know there's a very good chance it will be done in the future, and it has been done in animals. So this example here is of a dog which was genetically engineered to look like a lion. Okay, just kidding. This is from Purim. You can buy your costume there. But this is not a joke, okay? This is an actual dog which was genetically changed to develop muscles. So I don't see a lot of laughs now. And so that's another avenue in which like this might be useful. All right, so this is like what heritability is, like why you should care to make genomic predictions. And let's talk about the data format pretty quickly. So, okay. So the important thing to understand about DNA, and we'll go through this like pretty quickly, is that it can be understood as a really long string of letters. Okay, and our genetic information in particular is like split into 23 pairs of chromosomes. Okay, so you can look at like the top left for like one of the chromosome ones over there, and how do they look like? More closely, it looks something like this. So it comes in this data helix format, and after you like unzip it, it will look something like in the right. And just a little bit of terminology. So like each of the positions in the right over there, we call them a SNP. So these will be sort of like our feature names later, or like the column names, if you will. So we call that a SNP. So it's a specific position in a specific chromosome. And the value of that feature, or the value of that SNP, is an allele. All right? And each of these chromosomes, like depending on like which one it is, but in average it's something like hundreds of millions of letters long. So it's quite a lot. And it seems sort of like overwhelming. And but the good news is that sort of like despite all of our differences in in shapes and in colors, like humans are much more than 99% genetically the same. So if you take a random like set of like two people and you compare like their specific position, there is much more than 99% chance that they will have the same position. So one of the things it means is that you only need to sample a small fraction of your genome. So this is good because there is like less data. And it's also good because it's cheaper, and you're still preserving most of the information. So, but this will still sort of like easily like lead you to like sort of like tens of millions of features, which are not like as sparse as I would like, and that's sort of like the reality here. All right, so let's just talk quickly as well about how the data, how you get like from your physical DNA to data. And so the way it works is that you first like you get a kit, okay, and then you like swab your cheek for like cheek cells, and you put it in a tube. Then that tube goes to the lab, and then that your DNA gets put on this specialized chip, okay, and you can see like in this chip there that it has like 3 different fluorescence patterns. And these 3 different fluorescence patterns, they map to like 0, 1s, and 2s in your data, okay? I'm skipping like all the details here, but like the important thing to understand is that it somehow becomes like a matrix of 0, 1s, and 2s where you have, so like one chip is one person, so that would be like one row in your training matrix, if you will. And what do the 0, 1s, and 2 mean? So as I said, like the allele is the specific value of that SNP. And like typically, like this, when you have a 0, that means that like both of your chromosomes have the more common value at that specific SNP. If you have a 2, that means that both of your chromosomes have the least common value at that position. If you have a 1, that's one of each. All right, so that's so your data is like this matrix of 0, 1s, and 2s of like possibly like tens of millions of columns by whatever how many people you have. All right, so let's talk about some of the methods now. So we'll start by talking about some of like the previous methods, okay? And in particular, they mostly like boil down to something like a linear model, okay? And it's a very big like simplification, but like the reason why I'm comparing against linear models in particular is because they were, as far as I'm concerned, the ones that have been best able to predict like height, which was what we've been comparing against. So linear model is like what you guys know as, it's like you have like, that vector of like all of your SNPs, and you multiply it by another vector, maybe you add a constant, and you have your prediction in the right. So your sort of like polygenic risk scores coming from like GWASs, if you know what it is, like that falls in this context, as well as like something like Lasso, which has been able to predict like height pretty well, also falls into that. So, and what are some of the disadvantages of linear models? One is that it can capture something called like epistasis, which is just like genetic speak for nonlinear feature interactions. So it can capture a case in which like you only have an impact on a trait if SNP A and SNP B have a certain value, right? This can't be properly captured by a linear model or any other like nonlinear interaction. Another thing that it can be captured without further feature engineering, which can be done, but is not typically done in this context, is capture things like recessiveness and dominance. And another thing that it can capture is how covariates in fact impact effect sizes. So, for example, if we have, like, let's consider like the impact of like one SNP, and let's say that this like changes depending on like what subpopulation the person is from. Then, like without a lot of like other like feature engineering, this would not be able to be done as well. And yeah, like again, like just want to note, like this is like a big simplification of like other things, but since we'll be comparing it mostly against Lasso, that's like those will be like this, like the negative of that will be the advantages of our model. So. So I want to tell you guys about our like sort of like architecture at a high level. Okay, I can't mention too many like details right now, but hopefully like the high level picture will be clear and like what it can do. So basically, like at a high level, it is like a neural network, as you guys can see. So on the left, you have like all of your SNPs. On the very right, you have your height prediction, and there's sort of like two main like parts of the network. That's not too like surprising. In the first one, it's trying to capture sort of like lower level interactions. So this can be like individuals, like SNP effects. This can be things like epistasis and so on. And on the right, it's sort of like putting them all together and into like a fully connected layers and putting it into a really good estimator. And you know, again, like a few things to note. Like one, because sort of like so each of those arrows there, you can think of them as some combination of like a transformation with some regularization and a nonlinear activation and so on. So like because we have like a lot of those, and we are like those arrows, they are like placed in a way in which like it will like try to capture interactions where we think there are interactions. It can capture things like epistasis. It also captures like how covariates affect like low-level interactions. So as there is the arrow in the top there. So like we explicitly like feed in the covariates. So the covariates, again, they can be stuff like subpopulation, like statistics or gender and stuff like that. And because they are like at that level in the network, the network is able to distinguish between like impacts depending on like which subpopulation we have, which is something that can't be done in a linear network or in the previous methods. All right, so we'll talk about the results now. So the results, they are from a data set of 33,000 individuals of mostly European descent. Okay, so like trying to have a homogeneous population is a pretty like standard choice in these types of studies. And we'll be comparing it mostly against Lasso. All right, so Lasso is like linear regression with L1 regularization. And we're comparing against that because it is the method that has been able to achieve the best result in predicting height on the full, it's called like the UK Biobank. It's a data set of like hundreds of thousands of people. And they, as far as I know, like they've gotten like the best result in predicting height on them. So that's, we'll be comparing against them. And the R squared is sort of like the metric of choice in these things. So these are like our results. The blue is the like Lasso. The green is our method. And on the x-axis, you have like the training size, so ranging from 5,000 to 30,000. So yeah, I forgot to mention, out of the 33,000, 3,000 of those we leave for test set and we don't touch them. And the 30,000 we leave for training. And like one of the like nice things is that like it seems like sort of like this gap is not shrinking as the data set increases. So like the hope here is that like as we mentioned, like there is this like missing heritability in genomics, which is that like a lot of these traits, they're very highly heritable, but most algorithms haven't reached them. So Lasso in particular reached 40% R squared on the full data set of like 400,000 people. So the hope is that like once we get to that training size, this hopefully like will bridge some of that heritability gap that hasn't been explained. And on the right here, you see the true height. So this is like normalized and controlled for the covariates against the estimated height. And a couple of other notes about like sort of like spot checks like we did. One is that we compared learned features. So we compared like the linear coefficients in the lasso model against a measure of feature influence that we defined for the neural network. I'm happy to talk like later about how this feature influence works. And there's like a 0.5 rank correlation with the linear model. So it's, there's like a lot of similarities, but there's also a lot of like different things. And we also applied this model to totally like out of sample population. So we scored it on a data set of like a couple of thousand Ashkenazi Jews, which is like pretty like different from the training set. So it retained like a lot of performance. I think it's something like 13% R squared, which is I think a good result, given that it's not a training population. So these are the results. Like now, I just want to summarize and talk about like some other challenges and interesting questions. So I think like the main thing is that like these preliminary results showed that this neural network is outperforming linear models, and we really hope that like on the full like data set we'll be able to show that like it really outperforms also at that scale, which would be I'll be really happy if that happens. And some like other like future work to be done. One really important thing is to be able to generalize to different populations while preserving accuracy. So sort of like one fact about like most genomic data sets is that they contain like mostly people of European descent, either from the US or from Europe. But I think like if you're developing like models, you don't want these models only to work for like white people. So you want them to be like inclusive and like useful for everyone. So for example, like what can you do to bridge that gap? So like if your model, like given that it was constructed, let's say on the UK Biobank, which is like mostly like British people, can you take some like inner layer of the network and just train from that and make it work on like on another population that you might have access to? So this is like it's a really important question. Another direction is understanding like causality and mechanism. So like we think that this like sort of approach will be able to predict traits like better than like previous methods, but just predicting is not like it's not all we want. We also want to be able to understand mechanisms and like what's causing specific traits. So like which is something that unless you do like a lot of like side work with neural networks, is not you will not be getting for free. And another direction is multitask learning. So like. This sort of like has been done in genetics, not in the context of neural networks, but like training against different traits. But I think it's sort of like the perfect fit for neural networks. And so, for example, you can think of sort of like just training against like multiple like target functions of traits that might be related. So, and you can like imagine that some of the inner layers, they will be and they'll be able like to learn like, for example, like shared like mechanisms. And they will be able to like to regularize against each other and so on. So those are some cool like challenges ahead. We'll be working on some of them, and yeah. So that's pretty much all I wanted to cover. If this excites you, I suggest you take a look at these resources I have listed there for data. Or if this really excites you, I suggest like you also get in touch. We are hiring, and I'll be happy to chat more. Right.
Thank you very much. We have time for a few questions now.
Thank you very much. Two questions. First, are you obligated to GDPR? And second, what is the incentive for the individual to participate in such a survey or such a statistical model?
So the answer to GDPR is yes. Like I'm not specifically like assigned to that, but we have like a lot of people who are dealing with that. And so like then I think like for example I would have been I think there's something like nice about like taking part in a scientific study, and so like a lot of people like they volunteer there after having already done the test, like with the DNA test with us, they volunteer to take part in this study because
what is the restrictions in terms of third-party data?
Sorry, could you say it again?
What is the restrictions in terms of third-party data? I mean, what prevents you from agreement to have when you're not using my data with other external sources?
So I think like my heritage also, first of all, has been like around for a while. Like much before it has like a really good like reputation with its customers. It would be I think like this is not something that sort of like selling data. It's not something we would consider. Like if you're interested, I can check like legally exactly like why, but it's definitely not something that's on the table.
Additional questions?
Could you elaborate a bit more about the model that you used? Like, is it a standard CNN? Is it something else?
I can't like mention like too many details like right now, unfortunately, but it's not a standard CNN.
Do you know why you can't achieve the performance? The high bound, is it you don't have enough data, or it's about?
So I think like a large part of it is a part of it is definitely like data, but I think a part of it is just that so for us specifically, like it's definitely like data, because we're like at least like we'll be able to achieve like much more with more data. But if you're talking about like the linear model on the full like biobank data set that I mentioned, the 40%, I think there it's like partially like data, more data always helps, but it's also just that like the linear model like can't capture all those other things that we were talking about before.
Do you use any epigenetic features in your model at all?
No, we don't. That's an interesting question. So like like when you talk about like heritability estimates, this is talking about like all kinds of genetic components, and here we're talking about like we're only using DNA data. So there's also like perhaps like a world in which a large part of like the missing heritability is due to stuff which is not in your DNA proper. So I'm not sure if there's any good understanding in the literature about that.
We have time for one last question.
Hi. So you talked about the challenges of the generalization, and there is the no free lunch lemma, let's say. So let's say for other populations, how you handle it, actually.
And so I meant that more sort of like as a question like of interest. It's not really something that that we are working like really actively on. But I think like the hope there is that like, for example, let's say that you're talking about like the UK biobank data set, which contains like mostly British people, okay? And you have a model that works really well for them. And you want to like use that model, let's say for like another European population. Like I think it's assumed that sort of like a lot of like the learned features that were learned for like, that related population will also be like useful for the other one. So like even if you have like a smaller data set for the population B later, like some of like the learned features in model A will also be useful for that. So that's how I'm thinking about it. Yeah.
Okay, thank you very much, Luis. Take a break now.